Okay, for today's topic on the interactive case study 7, the description of the topic is Fluorocell, and the description is Fluorocell or 5-FU, sold under the trade name Adrucel, is used to treat various forms of cancer, including colon, breast, and digestive tract. Fluoropyrimidines were developed in the 1950s after the observation that rat liver cancer cells used uracil more rapidly than normal tissue. The combination of 5-FU with newer chemotherapies has improved response rates for advanced stage colorectal cancer by a dramatic 40 to 50%. Upon the uptake from the cell, the 5-FU is converted into three different analogs: fluorodeoxypyridine monophosphate (FDUMP), short name; fluorodeoxyuridine triphosphate, which is FDUTP; and fluoridine triphosphate, which is FUTP. Based off the presentation, the topic is walk the audience through how fluorophoricil is metabolized by cells in order to make a potent inhibitor. How To describe how fluorosil inhibits RNA synthesis, and to provide a mechanism for how this inhibition occurs. Um, fluorouracil 5U is a prodrug, which means it's a pharmacological inactive substance that is basically metabolized in the body, which then becomes an active drug once it's metabolized in the body. In the body, fluorouracil is converted to the active metabolite called 5-fluoroxyuridine monophosphate, which is F-UMP, the proper abbreviation. The mechanism of action is fluorouracil and metabolites have different numbers of mechanisms of actions. It acts as an inhibitor of thymidylate synthase, which is an enzyme that catalyzes the conversion of deoxyuridine monophosphate, which is called DUMP, to deoxythymidine monophosphate DTMP. It is an important precursor to DTTP, and also I'll show a picture here later on. But it is an active pyrimidine antimetabolite. Fluorouracil is synthesized into the nucleotide 5-fluoro-2-deoxyuridine. This product acts as an antimetabolite by inhibiting the synthesis of 2-deoxythymidine. The carbon-fluorine bond is very stable, and it prevents the addition of methyl group in the 5' position. The failure to synthesize the thymidine nucleotide results in no production of DNA or RNA. Fluorouracil causes the disturbance of growth of skin cells. It usually causes death of the skin cells, which grow really fast and cause abnormal skin cells. It can be used mainly the treatment of this is used to treat scaly overgrowth of skin, which are superficial to the basal cell sarcoma. Additional information about this is 5-FU acts in several different ways, but its main principal way is the thymidylate synthase inhibitor, which is a TS inhibitor. It interrupts the action of this enzyme by blocking the synthesis of pyrimidine thymidine, which pyrimidine thymidine is a nucleoside, which is usually used for DNA replication. The thymidylate synthase methylates deoxyuridine monophosphate DUMP to form the thymidine monophosphate DTMP, and due to the limited in DTMP, the cancer cell usually divides rapidly until it goes into cell death, which is why it's called thymidylase death sometimes, or most notably known as. Calcium folinate provides an exogenous source of reduced folinates, and which also stabilizes the 5-FU/TS complex. A little more information to add about this is that the main mechanism of FU's activation is conversion of the conversion of FDUMP, which is fluorodeoxyuridine monophosphate, and which inhibits the enzyme thymidylate synthase, which is TYMS. It also plays an important role of the folate homocysteine cycle in purine and pyrimidine synthesis. The conversion of the 5-FU to FDUMP can occur through the thymidylate phosphorylase, which is TYMP, to fluorodeoxyuridine, FUDR. And the action, and then after that, it occurs on the action of the thymidine kinase to FDUMP. And indirectly via the fluoridine monophosphate, which is FUMP, or the fluoridine, to fluoridine diphosphate. And then the ribonucleotide reductase action to the FDUDP and FDUMP. Then from the FUDP and FDUDP can also be converted to FUTP and FDUTP. And then this is also incorporated into RNA and DNA, which are the certain dynamics of actions of the fluoropyrimidines. And also want to show a picture of Fluorocell and the effects of it, which this is the 3D figure of it. Well, it's not really 3D, but 3D figure of the fluorocell structure. And this is like a little picture I found on the internet. It says the phases of fluorocell effects, early inflammatory phase. Just, more information about it is during the first week of application, mild inflammation occurs, as you can see. And then the second stage is inflammatory phase, which is followed by weeks of redness and swelling occurring, such as burning and crusting. And then the third is the tumor disintegration phase, which lesions resolve as the skin exfoliates. And the last phase is healing phase, where 1 to 2 weeks new skin grows into the treatment of the uric. Okay, thank you.